It's time for the 3-question warm-up for Renal 1. Let's get going. What are the manifestations of crest scleroderma? So remember, crest stands for something. It stands for calcinosis, Raynaud phenomena, esophageal dysmotility, sclerodactyly, and telangiectasias. Next question. What is the mechanism of action of penicillin drugs? Well, this is a cell wall inhibitor, so it binds penicillin binding proteins. It blocks transpeptidase cross-linking of the cell wall, and it can activate autolytic enzymes. So it is basically breaking up that cell wall, therefore it's considered bactericidal. Next, which embryologic structure of the heart gives rise to each of the following adult structures? So smooth parts of the left and right ventricle is the bulbus cordis. Smooth part of the right atrium is the right horn of the sinus venosus. Trabeculated left and right atrium is derived from the primitive atria. Trabeculated parts of the left and right ventricle is derived from the primitive ventricle. All right, that's it for the warm-up. Let's get to that lecture now.
Here we are again, and here I am wearing my yellow shirt. Now, when I wear my yellow shirt, I can only think of one thing: urine. So let's talk nephrology. In this video, we're going to talk about some basic renal anatomy. But first, let's talk about renal embryology. The kidney starts off as structures called the pronephros, the mesonephros, and the metanephros. But all three of those don't make it to adulthood. The pronephros is present at week 4, but then it gradually disappears. The mesonephros basically functions as a fetal kidney during the first trimester, and then it becomes part of the male genital system. And then the metanephros gives rise to the adult kidney. It begins to appear during the 5th week of gestation, and then it continues to grow and develop into the kidneys until well into the 3rd trimester. Now, the caudal end of the mesonephros, the end closest to the feet, develops something called a ureteric bud, and this gives rise to the entire collecting system, from the collecting ducts and the calyces and the renal pelvis all the way down to the ureters. Now, this ureteric bud also induces the mesenchymal tissue of the metanephros to differentiate into the renal tubular epithelial structures like the nephrons. So it's kind of like how the mesodermal notochord induced the ectoderm right above it to form the neural tube. These tissues interact with each other and induce the formation of new tissue. Now, what would happen if those ureteric buds failed to develop? Well, you wouldn't have a collecting system, but just as importantly, you wouldn't be inducing the metanephros to develop into the nephrons. So essentially, without ureteric buds, you're going to get bilateral renal agenesis. The kidneys fail to form at all. So bilateral renal agenesis results in something called Potter sequence or Potter syndrome. It's really a sequence of events. And Potter sequence is definitely high yield, 3 or 4 stars. So without kidneys, the baby's going to have oligohydramnios. Amniotic fluid is basically fetal urine, right? So if there are no kidneys, there will be no urine and therefore no amniotic fluid. But amniotic fluid is important. Without amniotic fluid, there's going to be pulmonary hypoplasia and limb deformities and facial deformities like a flattened face and also wrinkled skin. Now, obviously, this constellation of findings could be seen with any type of severe oligohydramnios. It isn't specific to renal agenesis. And really, the term Potter sequence is kind of an outdated name. Now, that said, there's a handy mnemonic for the features of Potter sequence, which happens to spell out Potter. So P is for pulmonary hypoplasia, O is for oligohydramnios, the 2 T's are for twisted skin, for that wrinkled skin, and then twisted face for the facial deformities. The E is for extremities, for the limb deformities, things like club foot, and then R is for renal agenesis, which is potentially a cause rather than a result of Potter sequence. And I find that Potter mnemonic pretty helpful. And then one other developmental anomaly to discuss is called a horseshoe kidney, which is also 3 or 4 stars. A horseshoe kidney is where the inferior poles of the kidneys fuse, forming a single kidney in a U shape or horseshoe shape. And then as this U-shaped kidney ascends from the pelvis during development, it's going to get hung up under the inferior mesenteric artery, and it's going to stay low in the abdomen. So, which artery traps the horseshoe kidney? The inferior mesenteric artery. Make sure you know that. And the other thing to remember is that horseshoe kidney is associated with Turner syndrome. All right, let's move on to some basic renal anatomy. So blood flows into the kidney through the renal artery, and it goes out to the peripheral parts of the kidney. And the peripheral parts of the kidney are called the cortex, and that's where the glomeruli are found. Now, the glomerulus is basically a tangled tuft of capillaries, and it's connected to a higher pressure afferent arteriole, which brings the blood in, and a lower pressure efferent arteriole, which carries the blood away. So the glomerulus accepts the afferent arteriole, and the blood goes through the capillary tuft, and then the glomerulus emits an efferent arteriole. Now the tuft of glomerular capillaries is encapsulated by Bowman's capsule, which I think of as like a little plastic baggie that contains both the glomerular capillaries and the filtrate that comes out of the capillaries. The part of Bowman's capsule that's up against the capillaries forms part of the glomerular filtration barrier. Now if you think way back to the beginning of the course in neurology, we talked about how the blood-brain barrier was made up of 3 layers: capillary endothelium, basement membrane, and the astrocyte foot processes. Well, the structure of the glomerular filtration barrier is very similar. There's the capillary endothelium, which in this case is a fenestrated endothelium, unlike the blood-brain barrier, because here we want fluid to leak out of the capillaries. There's a basement membrane, and in the kidney this is negatively charged in order to keep proteins from leaving the capillaries. And then instead of astrocyte foot processes, you have podocyte foot processes. Those podocytes are part of Bowman's capsule. We call them the visceral layer of Bowman's capsule. So blood flows into the glomerulus in the afferent arteriole, and then it circulates through the capillaries. And while it's there, plasma and electrolytes and urea and glucose and amino acids can all ooze through the fenestrations in the capillary walls and between the podocyte foot processes. And all that fluid ends up inside the baggie that was formed by Bowman's capsule. And we call the inside of that baggie Bowman's space. And from there, this fluid, which we call the filtrate, it's not called the urine just yet, from there this filtrate enters the proximal convoluted tubule, which is the first part of the nephron. And we'll pick up the flow of the filtrate again in renal 3, and we're going to follow it through the nephron as it turns into urine. Dress appropriately. But for the moment, let's go back to the blood that remained in the glomerular capillary. We said that fluid and glucose and urea and electrolytes can all ooze out of the glomerular capillaries. But bigger things like blood cells and platelets and large proteins, they're too big to pass through those fenestrations, so they stay in the capillaries, along with some of the fluid. And eventually all that stuff exits the glomerulus through the efferent arteriole. Now in the next video we'll talk about some of the things that either vasoconstrict or dilate these afferent and efferent arterioles. But that higher pressure afferent arteriole is pressed right up against the distal tubule of that same nephron. And right there in between them are some cells called juxtaglomerular cells, which means next to the glomerulus. Now this is part of the juxtaglomerular apparatus, or the JG apparatus. The JG apparatus is made up of 2 main cell types. You have the JG cells, which are responsible for secreting the enzyme renin, and you have some specialized sensory cells in the wall of the distal tubule right up against the JG cells called the macula densa, or the dense spot. Now the macula densa senses sodium levels in the distal tubule, and if the sodium level in the lumen of the distal nephron gets too low, the macula densa tells the JG cells to release renin. So there are 3 main things that tell the JG cells to release renin. One is beta-adrenergic stimulation. The second one is low sodium in the distal convoluted tubule, which is what that macula densa is monitoring for. And the third is low pressure in the afferent arteriole of the glomerulus. So the JG cells are actually monitoring the blood pressure, and if it drops, that's a signal to release more renin. So in the presence of high sympathetic tone or low sodium delivery to the distal tubule or low renal arterial blood pressure, you're going to have more renin released. Now, we've talked about renin several times already, but just to review, renin cleaves angiotensinogen into angiotensin 1, and then angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. And then angiotensin 2 has 2 functions. It tenses angios, it's a potent vasoconstrictor, so that raises the blood pressure, and it stimulates the adrenal glands to make more aldosterone. And then aldosterone causes sodium and water retention. So if the renal arterial pressure is low, the JG cells will sense that and they're going to secrete renin. That's going to ultimately generate angiotensin 2 and raise the blood pressure. Or if the macula densa senses low sodium in the filtrate, that means that there's low sodium in the blood. So the macula densa tells the JG cells to secrete renin, which ultimately leads to more aldosterone and more sodium reabsorption. We talked about how filtrate starts in the glomerulus and enters the nephron. And the nephrons form the medulla of the kidney. So the glomeruli are found in the cortex and the nephrons are found in the medulla. We're going to talk about the nephron in renal 3. But ultimately, the nephrons drain into the collecting tubules, and the collecting tubules drain into the medullary pyramids, and then into the renal pelvis, and that empties into the ureters. The ureters run retroperitoneally down toward the bladder, and they join the bladder at the ureterovesicular junction, or the UVJ. And it may be worth knowing that the UVJ is a common site for renal stones to get stuck. Then one other important point about the ureters is that they pass under the uterine artery in women, or under the vas deferens in men. Now, I'm sure you remember the phrase water under the bridge. So the urine is the water. The ureters are carrying the urine, carrying that water. They run underneath the vas deferens, or underneath the uterine artery. This is relevant because the ureters can commonly be injured during pelvic surgery. So say, for instance, you're doing a hysterectomy. You don't want to accidentally ligate a ureter thinking it's the uterine artery. Unlike in anatomy books where the arteries are nice and red and the ureters are gray or brown or whatever, in real life, everything tends to be the same pinkish tan color. So it's very helpful, very useful to be able to identify the ureters. So the uterine artery is the bridge, and the ureter is full of urine or water, and the water is flowing under the bridge. So now it's time for the end of session quiz. Go ahead and answer those, and then we'll go through the answers. All right, first question. What artery prevents a horseshoe kidney from ascending into the abdomen? Definitely important for you to know. It's the inferior mesenteric artery. Next, what fundamental problem creates Potter sequence? It's oligohydramnios, classically from bilateral renal agenesis. And what causes that? Failure of the ureteric buds to form. But the fundamental problem causing all the features of Potter sequence is oligohydramnios. Next, what cell type releases renin? It's the juxtaglomerular cells, the JG cells. And our last question, what are the 3 stimuli that induce renin release? Low renal arterial blood pressure, so low pressure in the afferent arteriole, which is monitored by the JG cells. Low sodium in the distal convoluted tubule, which is sensed by the macula densa. Then the third stimulus is beta adrenergic stimulation, or sympathetic tone. So that's the end of the end of session quiz, and that's the end of Renal 1. We'll see you next time.
Good evening. The annual Plaid Pants Festival went off without a hitch this weekend. Or did it? But first up, tonight Jane has a riveting story about the effects of Potter syndrome. Jane?
Thank you, Cliff. Potter syndrome has been a serious condition since the late 90s, affecting young children and older nerds alike in an epidemic fashion.
Jane, what are you doing?
I have immersed myself in Potter mania, Cliff.
Jane, I think you misunderstood the assignment. It's the features of Potter syndrome, not Harry Potter. Pulmonary hypoplasia, oligohydramnios, twisted skin, twisted face, extremities defects, and renal agenesis. Potter.
Oh, really? Then how is it that I'm riding this broom, Cliff? That's what I thought, Cliff. Now, if you don't mind, I am late for my Quidditch match.